In the distillation lab, you will distill a fermented batch of sugar solution. And because of a nasty little microbe called COVID-19, we are going to prepare that fermented batch for you. First step, place a plastic container on a top loader balance and tare it. The reading of the balance now shows the net mass of our sample, and we weigh out 50 grams of table sugar. Next, we weigh out 5 grams of baker's yeast, which contains the single-celled microorganism, the fungus that will perform the fermentation. We've weighed out 2 solid substances and now use a wide neck plastic funnel to transfer these 2 substances into a 500 milliliter Erlenmeyer flask. Next, we add warm water, about 40 to 50 degrees warm, until we have about 250 milliliters of total mixture. We then place the Erlenmeyer flask on a stirrer with a warm hot plate and let it stir for a couple of minutes in order to suspend the dead yeast cells and activate the living yeast cells to perform the fermentation. The metabolic processes that allow the yeast cells to convert the sugar into alcohol occur without oxygen in the atmosphere. It's called anaerobic. So to exclude oxygen, we place a rubber balloon on top of the Erlenmeyer flask. A byproduct of the fermentation is carbon dioxide gas. When you bake something with yeast, it makes your dough rise. In our case, the carbon dioxide gas inflates the balloon. For the distillation, you need about 100 milliliter of the fermented batch, which you will decant into a graduated cylinder. Decanting means to slowly pour it without disturbing the solid at the bottom of the Erlenmeyer flask.